Hi, I'm Kim, and I'm an Advanced Research Associate at Bio-Techne Spatial. I will present my poster entitled Spatial Immune Profiling of Human Colon in Health and Disease, Insights into Inflammation. The goal of this work is to demonstrate you how high-plex spatial technologies enable detailed mapping of immune and tissue architecture in healthy and inflamed human colon. The human colon contains a complex immune environment that is being remodeled in inflammatory bowel diseases. However, conventional techniques to explore this remodeling often use the spatial context. Here we leveraged COMET-based multiplex staining and imaging to profile immune phenotypes at single-cell resolution while preserving tissue architecture. In the first figure, you can see the COMET suite workflow, starting from tissue preparation outside of COMET. Then, sequential immunofluorescence cycles, consisting of iterative staining, imaging, and elution, are automated on COMET. Users can detect up to 4 markers per cycle on 4 tissue sections simultaneously. Then when the run is finished, COMET images are pre-processed and ready to be analyzed. Our Horizon software is actually tailored for COMET multiplex images, and this schematic represents the typical workflow to analyze SEGIF images with Horizon software. Here we developed a 63-plex protein panel to study multiple cell phenotypes, activation states, and morphological features of the colon from the same tissue section. 2 consecutive runs were co-registered by the integrated feature on COMET software to obtain a unique multiplex image with 63 detected markers. Table 1 shows the panel composition. Targets are classified by lineage and morphological features. This panel is built using Spire antibody panels and tumor-specific antibodies, as well as R&D system antibodies, including Novus, tested on COMET. So after the workflow, we show the TMA used in this study and how tissue morphology is preserved with the COMET run. The TMA used contains cores of healthy colon samples, as well as cores from patients with different pathologies, such as ulcerative colitis, colon disease, and others. Multiplex staining, H&E, and immunofluorescent images demonstrate consistent morphology and signal quality, demonstrating that the COMET is a suitable platform for studies where tissue preservation and accurate multiplexing are of paramount importance. We then looked at distinct immune populations such as T cells, B cells, macrophages, and neutrophils. These populations are identified and visualized spatially to perform high-plex dimensional immune profiling in inflamed colon samples. Importantly, inflammatory regions show altered immune composition and localization compared to healthy tissue. Next, we examined epithelial remodeling. Using Horizon, I performed the rule-based cell classification to identify epithelial cells as CK positive, and we observed that a reduced expression of CD138 and CDX2 was accompanied by an increased HLA-DR expression in ulcerative colitis and Crohn's disease, indicating epithelial remodeling and immune activation in these patients. Next, we analyzed immune activation by quantification of granzyme B positivity and Ki67 positivity in cytotoxic T cells, where multiplex imaging revealed increased activation of these T cells in inflamed tissues. Particularly in chronic disease samples, we saw the highest granzyme B mean intensity within the granzyme B positive cytotoxic T cell class. Finally, we investigated the spatial composition of lymphoid aggregates using unsupervised clustering in Horizon. This analysis reveals that these enriched and organized immune dishes may alter their composition during chronic inflammation, highlighting spatial organization as a key feature of intestinal immunity. In conclusion, spatial immune profiling using COMET reveals remodeling of immune architecture in the inflamed human colon. These insights would be missed using dissociative approaches, so this work demonstrates the power of high-plex spatial imaging for studying human disease and identifying potential human targets. Thank you for listening. Catch you in the next one.